Hi everyone, welcome to the 6th tutorial in our Imaging Science series here at the MIT Museum. For today's video we have a special guest, Jeffrey Kuhn, who is the Scientific Director of the Koch Institute's Microscopy Core Facility here at MIT, and he's going to discuss what exactly is a digital image and how we can use that knowledge to make adjustments to those images and to show things that we wouldn't have otherwise seen. And with that, I'm going to hand it over to Jeffrey.
In this video, we're going to be talking about scientific images, some of the most important features of them, and how you process them. I'm going to give you a little bit of background information first on what scientific images are and some of the basic features that we are going to need to understand in order to bring out more information from these images. So what are the main things we look for in a scientific image? Well, the first thing we look for is whether or not you can see what you want to see. And this is dictated by the contrast, whether something is visible in the first place. We also want to know if we have enough information to distinguish things, and that's resolution. And we typically think of resolution in terms of megapixels in your camera, but there's a scientific definition of resolution, and that is, can you distinguish two things as separate or not? There's also how much uncertainty there is in your measurement, how clear your image is. And every measurement you take has uncertainty. If you measure the temperature over the course of a day, your uncertainty might be a few degrees Fahrenheit. So images are no different. There's an uncertainty in the measurement of intensities of an image. And in imaging, we use the signal to noise ratio. If noise is the uncertainty in your measurement, the signal is what you want to see, we tend to measure the ratio between those two things to tell us how clear our certainty is. And then finally, there's the speed in which you take an image. That's called temporal resolution. So did you capture the right moment in time? So you can't have good contrast, good resolution, good clarity, and fast speeds all at once. There's always a compromise between these factors. There's always a balance you have to make. And this is called the triangle of compromise. So for instance, if you want something that's both bright and high resolution and detailed, you'll have to image very slowly. If you want something that very fast imaging and high resolution, you won't be able to collect as much light. It's going to be dimmer. If you want something that's bright and fast, you're going to have to sacrifice spatial resolution. But you'll end up with a blockier image. We can, however, balance between these factors. For most cameras, you can set the exposure time for your images. So if you want to do a fast exposure to capture fine motion, you can do that. But you'll be getting a dimmer image, and you might have to sacrifice some detail. If you want slower images, you can make them brighter. Some cameras let you actually change the resolution of the image, basically putting a bunch of pixels together into big superpixels. And that actually increases the sensor area of the pixels and makes them brighter. So you can get, at the expense of lower resolution, you can get a brighter image. Or if you want higher resolution, you're going to have to deal with dimmer or less light collecting ability. Also, you can balance between these things by expense. So bigger sensors and bigger lenses can collect more light, but that ends up being more expensive. So the camera equipment that you use can dictate how far along this triangle of compromise you can go. So a digital image is fundamentally a matrix of numbers that describe intensities. So we're going to use this image of an old world ape mandrill to talk about the numbers that make up the intensities in an image. And we're going to ignore color for the time being and just look at a black and white version of this mandrill. So let's take a look at a little tiny portion of the muzzle near this little red line on the muzzle. And if we blow up that little portion, that looks like a bunch of gray squares. And those gray squares are made up of a bunch of numbers. And if you look, the squares on the left-hand side tend to be a little bit higher value, let's say 180, 200, things like that. And those images are brighter. Those pixels are brighter. The ones on the right-hand side are a little bit lower value, 111, 118. They tend to be darker. So the numbers in this matrix describe intensities. So how do computers represent numbers? Well, we represent numbers the Arabic way, or in powers of 10. in decimal digits. So for instance, a 3-digit number has a ones place, a tens place, and a hundreds place. And for example, the number 136 is a 1 in the hundreds place, a 3 in the tens place, and a 6 in the ones place. You can sum all those together, 100 plus 30 plus 6, and that gives you 136. Well, computers represent numbers in a binary fashion. So they use a single number between 0 or 1 to describe a number, and each of these bits can go from 0 or 1, and they have different places, a 1's place, a 2's place, a 4's place, etc. So for instance, the number 1 can be represented by a 1 in the 1's place and 0 everywhere else. The number 136 can be represented by a number 1 in the 128 place and a 1 in the 8th place, and that adds together to be 136. If you have all ones in one of these 8-bit values, the maximum number is 255. So 8 bits can have a maximum of 255 and a minimum of 0. And it turns out the more bits you use in one of these images, the more shades of gray you can represent. So for instance, 2 bits can represent 4 different shades of gray. And so here's the mandrel with 4 shades of gray. You can also use 4 bits or 16 shades of gray, and now we're starting to get a little bit more detail in this mandrel. It doesn't look quite so chunky. 8 bits gives you 256 shades of gray, and now this mandrel looks very smooth, very good detail in this mandrel. But computers and sometimes scientific images can actually take even more than 8 bits. They might have 16 bits of images, of numbers in your image. So 65,000 shades. Or you might use color, where in most cases, most computers will use 8 bits per color. So 256 shades of red, 256 shades of green, 256 shades of blue. Well, let's look now at the difference between 8 and 4 bits. Let's zoom up on a little bit of a portion of this. Now, if you notice in the 4-bit image, you can see the gradations from gray to white in the muzzle. Whereas with 256 shades, you can't really see much of a gradation at all. It looks much smoother. So it turns out somewhere between that 16 shades and 256 shades is about where your eyes can distinguish individual shades of gray. So what makes a good image? These are examples of cells taken by microscopes. And most of these have slightly different contrast, and it's the contrast that makes them better or worse. So for example, the cells on the top left-hand side, the top left and the top middle, are taken with the same microscope using different contrast methods. In this top left image, you can barely start to see some cells, and in the middle image, you can actually make them out because they've got higher contrast. Similarly, this image of a cell on the right-hand side has pretty good contrast as well. Not quite as good as the one in the middle. And then in the bottom row, you're seeing good contrast between the 2 bottom images. You're getting nice, these are called microtubules. They're very bright against a dark background. And the one in the middle is less blurry than the one on the left-hand side. So blurring, it makes a little less or worse contrast. And then finally, color can add additional detail of contrast for your eye. So let's talk about contrast. Contrast is whether or not you can see something in the first place. So I've drawn a bunch of dots on this slide, and those represent a signal that we want. And I've put them on top of different backgrounds. And the background is what we don't care about or what we don't really want. So I've increased the signal from going from left to right-hand side, and I've increased more and more background going from top to bottom. So let's take a look at the left-hand side, the weakest signal. You can actually start to see this weak signal on top of a black background. But when you've got a really bright or white background, that signal is almost gone. And it turns out the definition of contrast is essentially the signal you want minus the background you don't care about divided by the background. So, but intuitively, what we think about is that the more background you have, the weaker the contrast. The more signal you have, the higher the contrast. So the contrast increases from left to right as we get more and more signal, and it increases from bottom to top as we get lower and lower background levels. To give you an example of how background can affect contrast tremendously, stars are actually out during the day. You just can't see them because the sunlight is so bright and the sky is so bright. But once that brightness goes away and day turns to night, the stars that were the same signal they were before are suddenly visible because the background is no longer there. So background can affect contrast tremendously. Now that we understand contrast, let's talk about what types of intensity values we're going to display from an image. So most monitors and printers and computers store images as 8 bits of intensity. Higher grade scientific cameras, or if you take a high dynamic or range or HDR photo, you might store those images as 16 bits or 16 bits as raw data. But most computers can only display 8 bits of information at any one time. So what ranges of values of that 16 bits are you going to actually choose to display? Well, if you have something that's really bright, you might choose the topmost bits or the brightest bits to display and scale those to 8 bits that your monitor or computer can display. If that image is fairly dim, you might take the dimmest pixels in that image and stretch them into the 8 bits that your monitor can display. But that same sort of transformation can also apply to 8-bit images. We can take a range of those values from an 8-bit image and choose which range to display on your monitor. So in order to understand what we're doing with this, let's talk about how we analyze image intensity. So there's actually a way to plot all of the image intensities in your image, and this is called a histogram. It's a plot of intensity values. So what we're going to do is take our mandrel example and that matrix of numbers from that little tiny portion of the muzzle, and we're going to count the intensities by range. So let's take the range of pixel values from 100 to 119. If I count those out, go through all that and count out, I get about 10 pixels from that range. If I now go from 120 to 139 and count all those up, I get 13. If I count from 140 to 159 intensity values, there's only one of those, so it's a very small number. From 160 to 179, I've got about 19, and et cetera, et cetera. You can continue counting in these binned ranges. Okay, so what does that give us? If you look at those counts, those tally marks, that looks kind of like a graph. Turn that graph sideways and plot it out for the entire image, going from low intensities on the left, or 0, all the way up to 255 for the highest intensity, and we're going to plot the counts as a graph. And this ends up being called a histogram plot of all the intensities. So it's counts over the entire range of intensities that are possible. So a histogram is actually a very informative plot of what kind of intensities are in our image. So for example, for this histogram, we can see that there are very few black pixels and there are very few white pixels. And the majority of these pixels are mostly gray, centered right about the center of this image of the intensity values. Let's take a look at another image. So this is an image of a bunch of boats, and we can see that there is a bunch of high number of black pixels. So those black pixels correspond to the bottoms of those boats. And there's also a bunch of gray pixels that correspond to the sea and the air and most of the boat, and a few little whiter and brighter pixels as well. But this gives us an indication of what kind of intensity values our images contain. Even color histograms can tell us something. So I can take all of the intensities in the red channel, the green channel, and the blue channel and plot those individual histograms. And so, for example, in the highest values at 255, you see a lot of red and just a little bit of blue and no green. And those correspond to the bright red stripe going down the center of the muzzle of this mandrel. Just a little bit below those, there is some fairly bright blue with no green and a tiny little bit of red, and blue and red together make up magenta. So this is the magenta and blue stripes along the muzzle next to the nose. A little bit farther or dimmer, you can see a bunch of green and a little bit of red and very little blue. And this corresponds to the yellow whiskers because red and green together make yellow. So we can get a little bit of information from color from a histogram, and you can do the same sorts of plots of intensities of color. So how do we use these histogram plots to bring out information from an image? Well, let's draw a line going from the bottom left to the top right corner of this histogram. And we're going to use this original histogram as the input, and we're going to create an output histogram corresponding to the image that we want. So the bottom, or 0, in the input corresponds to 0 in the output, and 255 on the input corresponds to 255 in the output. And we call this transformation a linear map. We're going linearly from 0 to 255 on the input, and that maps to images or values from 0 to 255 on the output side. But what happens if we stretch this line or change the slope of this line? So let's take the minimum and raise it up and the maximum and lower it down. If we stretch that image, we essentially are narrowing our contrast range to what's actually in our image, to the values or brightness values that are actually in our image. So we can actually make a better contrast image of this mandrel. But we don't have to stick to a line. We can also use a curve to describe a transfer function from input to output. So you can use a curve to the bottom right, for instance, to get rid of some of the darker values while keeping some of the brighter values. Or you can use a curve to the upper left to boost the darker values without really increasing the bright values as much. So we can use these tools, the histogram and these transfer functions in our minimum, our maximum, and our gamma to bring out information and to get the most out of our scientific images. So let's use our newfound knowledge of image intensity values and histograms to try and bring out some more information from some scientific images. So we're going to be using the free image manipulation program called GIMP. It stands for the New Image Manipulation Program. But most of the other software packages you might run across will have the same sort of histogram manipulation screens that you can use to bring out intensity information and hidden information from scientific images. So I'm going to use GIMP, and I'm going to drag one file onto this image manipulation program. Let's open it up. So what we have here are a bunch of stars taken against a black mountain. All right, so let's see if we can bring out some other information from this particular photo. So we're going to go to the main histogram window for adjusting and doing these maps. So there are two that we want to talk about in GIMP. We're going to talk about under the colors menu, there's something called levels and there's something called curves. So these two menu items right here will let us manipulate the histogram. So let's start with levels. Okay, so levels shows a histogram of the intensity values within this image, right? So you can see this is our histogram. We're going from dark pixels to bright pixels, and we're seeing a count of dark to bright pixels. Now let's take a look. There are two basic humps, and the first thing I'm going to do is take this maximum value and drag it down all the way to about where we start to see stuff. Well, guess what? Now we can start to see we've got these dim stars in the background, and by just changing the intensity levels, we're able to bring out some of the information that was hidden to our eyes in this image. So we can now see the Milky Way, we can now see more more stars, and we can start to see this mountain right here. Let's bring the intensity down a little bit more. Okay, now the stars are really bright, and now we're starting to see a little bit of this mountain. So it looks like if we look at this histogram, most of this most of this comes from the mountain, and most of this hump comes from the stars. So we could bring out the stars at the expense of the mountain. And remember, if we drop the background down, we can get a little bit better contrast. So if we want to focus on the stars and the Milky Way galaxy portions sitting right across your image, we can bring the bottom a little bit up, and that decreases the apparent background. So I'm just going to zoom in. I'm going to hit plus on the keyboard, and we can start to see individual stars. All right. So we brought out some hidden information in this image. Let's take a look at a couple of other images. So let's take a look at a daytime image. And let's see if we can actually see some stars in this. And I bet you, oops, let me get rid of this. Start changes. Let me open it up. Let's see if we can see anything inside this image. So I'm going to bring up, again, colors, levels. And right now, this bright stuff here is showing the sky and the brightest portions of the image. But this histogram is really, really dim. It's really hard to see because the counts are linear. There's a little button up here, and a lot of your software programmers will have something like this, and it makes up what's called a logarithmic histogram. So this basically expands the histogram out so you can see more of what's happening. So again, now this logarithmic histogram, if I drag down, great, okay, so we can't see anything at the dark because the bright image is just too bright. Okay, so we're not going to really be able to pull out. Let's try dragging the bottom back up. Okay, let's change the background, to lower the background. Oh, we still can't see stars in this image, and that's fine. It's too much, too little contrast in our original image. But let's do one more thing. Let's go to colors. Let me reset right quick just to get back to normal. And I'm going to go to color curves. Okay, so this, remember that transfer function we talked about? Well, we can decrease or increase the minimum and turn a little bit more black. You can also change to a curve. So we could potentially make this mountain a little bit more contrasty by changing the intensity curve. And the fun thing about this is that you can actually put multiple curves on your color curve. You can put multiple inflection points. Okay, but now that we understand what's happening, it gives us a little bit of better grasp of what we're doing with these sorts of intensity transformation curves. All right, so let's see if we can bring out something else. Let's start with another stars image. So let's drag another image onto here. Okay, so now this image is from a 16-bit or raw format. So the previous image right here was only 8 bits. And let me take a look. Let's go to image precision, and this is an 8-bit integer image. Okay, let's take a look on this. This, if we look at image precision, this says 16 bits. So there's more information in this image than there was in the previous image. Okay, so let's bring up our color levels again and take a look. And remember, I still got, this is the normal histogram. Actually, let's go back to this linear histogram versus the logarithmic histogram. So I'm going to click on the linear histogram right here. Okay, so again, we're seeing two humps. One is a black hump and one is a slightly less black hump. So let's bring our maximum down, and again, we can start to see the stars, right? So we can see a lot of interesting things in here. And again, I can bring the minimum up and to just increase the contrast of the sky, of this night sky. So now we're seeing a little bit better contrast here of the night sky. But we're still washing this out a little bit. Let's drop this back down. So a little bit of sunset left at this twilight image. But remember this little hump right here? Let's move our minimum and maximum to span that hump. And guess what? Look at that. Now we see the mountain in essentially like daylight conditions. And the 16 bits of information, what that means is that we're getting a lot more shades when we're looking at this tiny little range of our input values. We're getting a lot more shades out because there's more information stored in this 16-bit image. Okay, let's take a look at another image. Let's see if we can pull out more star images from, let's say, an eclipse. So this is the sun during an eclipse, and this is not quite a full eclipse yet. And again, this image, if we take a look at the precision, this is an 8-bit image. So there might not be a lot hidden inside this image, but we're going to try anyway. Let's go to levels. And it looks like there's just a tiny little bit of information down here. Let's switch to a logarithmic histogram. In reality, this is logarithmic. There's not much information right here. But I can still drag this maximum down. Okay. And now the corona from the sun is peeking out a little bit more. We're seeing more of that. But in reality, we're not really seeing much in terms of stars. This image was just, the raw data was just too dim to get at the stars behind this image. So let's try another eclipse image that's a little bit farther along. Pardon me. Open it back up. Okay, so this is at a full eclipse, and now this corona is really bright because this image was taken at a much longer exposure. Still, it's an 8-bit image, but let's see what we can bring out with the levels. Okay, let's bring our maximum down. Okay, and now we're starting to see some things. And here's something else I want to point out. So we're starting to see some spots in the background, and I am going to have to zoom up to take a look at these. Okay, so, and I can hold down the space bar and use this to drag our image around. I can use the plus key to zoom in and the minus key to zoom out, and the space bar and dragging to come along. Okay, so now we're starting to see blobs right here. This is a star. I can zoom up on it. And I can tell that this is likely a star because it takes up more than one pixel. You notice in the background area right here, there's a bunch of noise, and this is what happens with low light images. Your cameras just generate a lot of noise, and that tends to be really low values of intensity, but it's still there. When you're blowing up the intensity like this, that noise comes right out. And you'll notice that there are single points of noise right here. It looks like individual pixels might be bright surrounded by darker things. And that's an indication that what that is noise, things that tend to look square. Whereas, do you see this right here, this blob? That is likely a star. This blob right here is likely a star because it takes up more than one pixel. It's taking up a few pixels. So that is a collection of photons, very dim collection of photons, but a collection of photons nonetheless that indicate that what you're looking at is a star and not noise. So again, there's less clarity to this image because the signal here is very weak and the noise is very strong. Okay, so now let's look at a biological image, something a little bit less astronomy. So this is an image of a flower. And let's see if we can find something else on this flower. So I'm going to go to color level. And let's take a look at our histogram. It's a little bit low, so I'm going to switch to a logarithmic so I can see more of the values right here. Okay, and typically what I do when I'm looking at a scientific image is I will play around with the minimum, and I'll drag that up and down and see if I can see anything new. There's something that's poking out, and I can drag the maximum down a little bit and see if I can see anything at the dim portions. Okay, so let's drag up this minimum, and you're starting to see something with a little bit more contrast. Looks like there's a caterpillar on this flower. And we can bring it out with just manipulating the histogram and this transfer function, the input versus output levels. Similarly, let's take a look at these petals. So I'm going to zoom up on a petal, right? And these petals are fairly dark. They're darker than the center of the flower. Let's zoom back out. So the petals here surrounding, and even this black space, the flower is really bright. But sometimes, if you're looking for something really bright in an image, it's going to overwhelm the dark stuff that you might be interested in seeing. So I'm going to drag this top portion down and see if I can bring out some detail at the darker levels. Now, at the darker levels, you're starting to see potentially pollen on this flower. Oh. And you can see the veins on the flower petals a little bit clearer. So sometimes, and let's just compare that to dragging all the way up. Okay, so sometimes the information you're looking at is hidden in dark information. And you can sacrifice bright pixels and bright portions of your data in order to look at the dimmer stuff. So that will hopefully give you a little bit of an idea of how to bring information out of scientific photos, to look for the hidden side in the images you took, and to understand what image intensity levels and histograms and these transfer functions can show you with scientific images.
Thank you for joining us for this tutorial in our Imaging Science series with the MIT Museum and Koch Institute. We'd love to see your stunning science photos. If you'd like to share them with us, you can share them on Facebook, Twitter, and Instagram, and use the hashtag MITMImagingScience to share those with us. At the end of this program, so on June 20th, we'll be holding a webinar to look at what makes a great science photo, and so submissions have a chance to be featured during that webinar. For more information on the Imaging Science program, other tutorials and other information and resources, please visit the MIT Museum website at the link below, and you will find a list of our other resources as part of the Imaging Science program.